Dear student, welcome to the basic genetics class, and we are now discussing the week 7 where we are talking about gene mapping on eukaryotic chromosomes. So, this is the lecture 2 of this week where we are going to discuss about the recombination and also cytological determination of crossing over. So, just to remind you where we were, so this week's classes are more exercise based. So, I think you all should practice the examples, and if you face any difficulty you can write me so that I can help you solve the problem, because this class is mostly about how to calculate the map distance, how to see how far or close two genes are. So, that can only be learned when you do more and more practice, and I would also go a little slow and keep you reminding what we have learned in our last class, so that you do not lose the pace. So, in my last class, as you can see on this slide, we learned that linkage modifies Mendelian ratios, right. So, we learned that Mendel told us that the genes they segregate independently, they assort independently, the alleles segregate and the genes independently assort, right. But when in the previous class we learned the concept that there are chances, actually there are many cases where genes do not really independently assort, right. And then that is the point when I introduced the concept of linkage, and I told you that the genes can show the linkage and they can be separated because of crossing over. So, crossing over provides genetic diversity even with linked genes, right. So, the genes are linked on one chromosome, but what we learned that because of crossing over, because of this breaking and rejoining of the chromosomes, we get new combinations. So, this is just to remind you, this is an image that we have used where I showed you that a chromosome, when it has two sister chromatids, means 4 chromatid state for 2 homologous chromosome, actually that is when they undergo crossing over. They do crossing over by involving 2 sister chromatids of the neighboring homologous chromosome, or more than 2 sister chromatids can also be involved. We have already discussed this point at length in our previous class. So, now I am trying to help you understand how these recombinations, means new combinations, are generated, right. So, you already learned, we have emphasized enough that because of the independent assortment we get new combinations, new genes have an opportunity to come together and show their phenotypes, right. So, we have Mendel's example, will not repeat it again. Last class I also showed you that we get new combinations even for the genes which are present on the same chromosome, but if they undergo crossing over, then we have another chance to get the recombination. So, this is an important concept note that crossovers are one mechanism for generation of recombination, and so is the independent assortment, right. So, they are both doing same thing, creating new combination, though the mechanism is bit different. So, let us take this example. Now, we are focusing on more and more on the examples where genes are present on same chromosome. So, if you look at the gametes here, you will see this capital A and capital B allele, they are together, and in the another gamete small a recessive alleles, they are together, right. This one, these small a small b, they are together. When we are putting this dot A dot B, this dot means that I do not know the order of the gene. It could be B dot A or A dot B. Once I know in which order they are, then I will just simply write them, A, B, or whatever way people usually write the genes. We will talk about that later. But this dot signifies that we still do not know their orientation, right. So, if you see that these A and B, when the gametes fertilize, they become the meiotic diploid, and they become the heterozygous, right. And when they undergo meiosis, we get the combination, right, the 4 combination. If we have 2 genes, then we have 4 combinations in a meiotic separation. So what combinations we are going to get? You see, because of this crossing over, what combinations we are going to get? One is AB. So now I want you to pay attention to this one concept which I am going to repeat in this class over and over again. What are parental types? Parental types, right? So what are parental types? Parental types are those that look exactly like the parental combination. So if you see A and B here is exactly the same that is present in one of the gametes. Same is true for small a and a small b, it is present in another gamete. So that is why we marked it as parental type. And the rest of the two, you see capital A with a small b and a small a with capital B. These are recombinant types, right? Why recombinant? Because this crossing over has taken place, and because of this crossing over, an exchange of fragment has been occur, and because of that now capital A is with small b, not with capital B, right? So this is the recombinant type. So this you need to remember always, from this point onward, always you need to pay attention. What are the parental type? What are the recombinant type? One more thing important that you should pay attention to, how much recombinant type we expect if genes are independently assorted, right? So this is something that we have to keep in mind very clearly, and that is come this example that I have put here, and you will see this symbol many a times on the slides, where I want you to pay more and more attention to understand the concept well. So if you see that the frequency of recombination in the set when the genes are independently assorted, it will be 50%, right? So it cannot go beyond 50%, that is what I am trying to say, it is 50%. If they are independently assorted, then you will see this as 50%. So just look at this example, and if you look at this example, what you will see, the same dominant combination cross with recessive combination, you get this heterozygous F1, and if you cross this heterozygous F1 with the recessive tester, then you will get these combination 1:1:1:1 ratio, right? So in one ratio you are going to get these two parental type, capital A, capital B together, small a, small b together. So they are the parental type, and these two, capital A with small b, small a with small b, are the recombinant type, right? So maximum possibility out of the combination that you have to get the recombinant is 1/4 and 1/4, right? So only half, which means 50%. You can only get recombinants up to 50%, right? So 50% recombination frequency will be seen in the case of genes located on separate chromosomes, remember Mendel, or far apart on the same chromosome. So now see another confusion I am adding here, because I already told you that two genes that are present on same chromosome are called linked. Now what I am saying, if they are far apart, consider them that they are segregating independently, right? So I will help you understand this thing better in just a few slides. Because we cannot say two genes are linked if their recombination frequency is 50%. You just saw why 50% is an important number, right? So those genes that are sufficiently far on a chromosome from each other, then we cannot call them linked, right? You got the point? So that means that they are unlinked technically, because they have all the possibilities to segregate independently. So now to avoid this confusion, what we used to say, two genes are said to be syntenic, right? Remember this word, syntenic, if they are on the same chromosome, regardless of whether they show independent assortment or linkage, right? So to avoid this confusion that linked genes or free recombinants, or if they are independently assorting even though they are on the same chromosome. If they are on the same chromosome, then we are using the word syntenic. Two genes present on the same chromosome, despite of their distance, they are close, they may be far, but we use the term syntenic. Remember this is important. So now let us learn how we are going to calculate the recombination frequency, right. So I am taking this example most of the time, same example in my slide, so that you do not get confused and you just have an idea of looking at the same thing again and again with and asking different questions using the same data. So this is the same cross that we have discussed, where we have these recessive and dominant combination. Here we have F1 heterozygotes, and with heterozygotes purple and vestigial, we are crossing them with tester recessive male. And this is the example we discussed in the last class. You can also go back and see that purple and vestigial, we are getting them in these numbers. So these purple and vestigial for wild type means, so these are normal, these are the wild types. So they are normal, and purple and vestigial, they are the mutants, recessive mutants, homozygotes, and these are the recombinants, purple plus vestigial and purple vestigial plus. And we did say that this is a number for them which is much less compared to what we expect. So it is deviating, it is a test cross is still deviating from 1:1:1, have already discussed it. Now using this example, we are trying to calculate the recombination frequency, right. So when we want to calculate recombination frequency, what we need to know? We need to know two things. One, the recombinants. So we need to identify this class. Remember I told you how we are going to identify the parental class? By comparing them with parents, right. If we compare this with parent, we will identify that this genotype is present as such in the same combination in the parent, that is why it is parental type. And when we are calculating recombination frequency, we need to identify the recombinants. Once you know that these are the recombinants, what you need? You need the total number. How many total offsprings are analyzed, right. So you have the recombinants, you have the total number, you divide the recombinants, total number of recombinants with the total number of progeny that we have, and multiply it by 100. And when you multiply it by 100, you get the recombination frequency in percentage, right. So now if you see this example, here we have the recombination frequency much less than 50%, it is only 10.7%, and that indicates that these genes are linked. So now one add-on concept, that when for us it is also very important what is the combination of the allele in case of parents, right. So in the previous example, or so far the examples we have covered except one in the last class, we always keep the dominant alleles on one homologous chromosome and we keep the recessive alleles on another homologous chromosomes, right. So this is one way, and you have already noticed, I will repeat it actually once so that it is clear to you, that we say that the parental types are predominant. So here the same example, I am not going to repeat it. Here you can see the dominant alleles are on one homologous chromosomes, both of the dominant allele and both of the recessive are on another homologous chromosome. Remember, we call this kind of configuration as cis or coupling. Mean both the dominants are together on one homologous chromosomes, you have both dominant or both recessive. That is the cis configuration or coupling configuration. Whereas if you compare this one, in this configuration you have a gamete where the capital A is with small b, one dominant is with another recessive, and one recessive is with another dominant allele of the like two genes. When we are looking at two genes, we have one allele for one gene dominant and another allele for the gene for another gene is recessive, right. So on one homologous chromosome, they are neither both the alleles are recessive or dominant, right. So this kind of configuration is called as trans or repulsion. So remember, many times this just thing being asked in context of terms, and sometimes even in solving the question, this understanding is very important in identifying the parental types. So now you see, how does that affect the parental type? Clearly it is. So you see, now the parental type will be A and small b, or small a or capital B. It is no longer capital A, capital B. Do not expect that in a numerical always you will be giving capital A and capital B together and small b together. You will get these kind of situations where you will get repulsion combination of alleles. So your parental change, and so is true with your recombinants, right. So this is important for you to remember the coupling and the repulsion configuration. And if you remember in the previous class, I did tell you the example of Morgan, where initially when he identified that the genes were not independently segregating, then he set up a cross with the heterozygous in the F1 progeny. And this is the same example, the same data that I have shown you in my previous class, I have repeated here. So you see, in this one example, you have these both dominant together, both recessive together in parents. So in this example, this is the F1, and in F1 dihybrid, you have this coupling parents PR plus VG plus one homologous chromosome and another homologous chromosome where it has recessive together, and this is test cross. So whereas in here in your F1 you have combination, you have PR plus with VG and PR with VG plus, and you are doing the test cross here. And if you see, you will see that because these two are the parental class, these are the predominant class here, and because these two are the parental class, these two are predominant class here. So for this one example, and if this is a two example, then for one example these are the recombinants, and for two example these are the recombinants. So I hope I have helped you understand the concept of identifying parental type and recombinant type enough, and probably you will not have any trouble in identifying them eventually. So let's talk about the coupling and repulsion just in a like a summary way, what exactly we learned. So coupling is a cis configuration, and in cis configuration all the wild type alleles are on one homologous chromosomes and all mutant alleles are on other homologous chromosomes. It's just for you to remember for the sake of your notes. And in the repulsion phase configuration, what we have that each chromosome contains one wild type and one mutant type of allele. So now we are coming to the next concept for this class. So remember, we mentioned that when Morgan saw these recombinants, he said that these genes are linked. But to understand or to tell why if they are linked they are getting separated, he said that it is because of crossing over. So that is his, he introduced this concept of crossing over, that the chromosomes can break and rejoin and exchange fragments, right. But it was not seen, right. It was theoretically, while he was conducting the experiment, looking at the data, conceptually he proposed it, that this is something which is happening, but how it is happening, what exactly it looks like has not been seen or understood that well, right. So if you even see, I mean I just thought of putting this image back from my very first class when I was discussing with you the work of Sutton. You could even see these homologous chromosomes pairing and even touching, making probably chiasma in these these kind of structure that he was seeing. So after him people have seen him even better, they have talked about it. But what is the significance and importance of these chiasma, the touching points for the chromosomes, was it took time to understand, right. So you can see 20 years it took after Morgan to really understand how exactly the chromosomes exchange the segments, right. So it's not just one evidence, but the two independent scientists using two independent model organisms showed that chromosomes indeed exchange their fragment, and that was the real visual evidence. Under the microscope, these people were able to show that phenotypically there are some traits that are associated with some chromosomal phenotype that you also can see under the microscope, and when they exchange these part of the segment that carry those genes, you see the phenotype and you also see the changes in the at the chromosomal level. So let us look at this one example. The first example, this is a work by Barbara McClintock and Crichton. So if I am not wrong, she was her student, and it was her remarkable observation. This Barbara McClintock and team is also very admirable and great, as you would admire many scientists. So she is amongst also one of those who did outstanding fantastic work in genetics. So well, in 1931, what did she see? She was able to identify phenotypes, as I just mentioned, with specific chromosome morphology. So see, she used cytological landmarks which was situated on chromosome number 9. So on chromosome number 9 there was a translocation near VAC gene. So endosperm composition is VAC or starchy. So VAC is dominant and starchy is recessive, and then the other gene is C, which is the seed coat. Capital C is dominant, which means that it is colored, and a small c is recessive and it is colorless. So you see, with VAC she saw this kind of attachment, right. So what are they? They are the translocation. On chromosome number 9, a part of chromosome number 8 was translocated. You will learn about these concept of chromosomal organization in other classes. So don't worry about that. We will discuss with you in very detail. But for now, you just understand this one point, that a fragment of one chromosome is getting attached to the other chromosome. So here on the chromosome number 9, you get this fragment of chromosome number A attached, that is one thing, and the other mark is this densely stained material near the seed color gene, right, seed coat color gene, right. So that is what here. So what she was analyzing, that if the recombination is taking place, then what would happen? This, right. So this recessive trait goes with this dominant coat color trait and the norm, right. So definitely now you can identify the norm is attached with the coat color, and you can also see that the recessive trait VAC is gone with the coat color, right. So you may not be seeing the recessive at such at that point until it is homozygous. So you can clearly see that the coat color is maintained and you lose the VAC endosperm composition, and same is the other way round. So other way round is what? Other way round is when you have this long attachment with the VAC, right, and it is going with the colorless seed coat, right. So this one, this combination. So you can see the phenotype and you can see the association of this phenotype with a chromosomal marker, right. So that is how she was able to clearly demonstrate that indeed when the crossing over takes place, chromosome exchange the segments. And similarly, almost on the similar line, but exactly the same time, 1931, Curlstone also showed, using the Drosophila, the fruit fly, that chromosome exchange their fragment. Almost similar strategy if you see. What he did is he used a gene which is bar eye. So usually the fly eye are bigger, but when they have this bar mutation, then they become very narrow, right. So, they are very narrow eye, and the other gene was carnation, which controls the eye color. So, what they see, so these are the phenotypes you can see in the fly, and along with these phenotype, what you can also see, you can see the visible chromosome abnormalities. What were those abnormalities? Actually similar. You see that there is one X chromosome, so these genes are on X chromosome. One X chromosome has a small additional translocation which is from the Y chromosome. So, part of Y chromosome get attached to the X chromosome, and the other X chromosome is shortened. You see this part is was gone. So, it is a deletion. So, this combination has a translocation as well as deletion, and by tracking that bar phenotype is associated with the small short X chromosome, and the other X chromosome has this where both the wild type alleles are present, have this Y chromosome translocation, and if crossing over takes place, then you start seeing that you have bar as well as the Y chromosome translocation. So, that is how using the this was the female, heterozygous female, and then they crossed it with recessive male with the CAR gene. So, that is how he end up identifying that crossing over indeed is a process where the chromosome break and rejoins, right. So, with that I will end here. You have 2 activity questions displayed on your screen. I am not going to read them through, you just go through them and try to solve the questions, and feel free to write your answer or doubts. Even if you have any trouble solving them, we will be available to help you with that. So, with that I will thank you all for your time and see you in the next class.